Hello, everybody. My name is Paul Wallace, and welcome to today's webinar titled Beyond Bulk Immunity, Dissecting Antigen-Specific B cell or B and T cell responses across species. I have the pleasure of introducing our webinar speakers, Drs. Jakob Zimmermann, Petra Bacher, and Tim Rollenske. Dr. Zimmermann is a group leader at the Institute of Systems Immunology at the University of Würzburg, supported by an ERC starting grant. His research combines microbial, notobiotic, and immunological approaches to understand how microbial-specific CD4 T cells maintain intestinal homeostasis and prevents chronic inflammation. His work has been recognized with several awards, including the Echo Young Investigator Award. Professor Dr. Petra Bacher is a professor of immunology at the University of Kiel, with appointments at both the Institute of Immunology and the Institute of Clinical Molecular Biology. Her research focuses on antigen-specific CD4 T cell responses in humans, particularly how T cells recognize microbiota and how these interactions can shift from protective to pathological in chronic inflammatory diseases. She's a recipient of the George Kohler Prize from the German Society for Immunology. And Professor Dr. Tim Rollenske is a junior professor at the Institute of Molecular Medicine and Experimental Immunology at the University of Bonn and the Emmy Noether Fellow of the German Research Foundation. His research integrates human samples, mouse models, and single-cell B-cell receptor analysis to understand how the humoral immune system shapes beneficial host-microbe interactions. His work has been recognized with several awards, including the Fritz and Yerso Mecker's postdoctoral prize. So really, we have a wide variety of systems looking at both human, T cell, B cell, and I believe also murine. And with that, we'll start, but be aware that you can put your questions into the Q&A box, and we'll be answering those at the end of the presentation. So why don't we go ahead with Beyond Bulk Immunity, Dissecting Antigen-Specific B and T cell responses across species.
Yeah, welcome to this webinar. My name is Petra Bacher, and together with my colleagues Jakob Zimmermann and Tim Rollenske, we will talk about Beyond Bulk Immunity, Dissecting Antigen-Specific B and T cell responses across species. So this is of course a broad field, and since we cannot cover everything in this short webinar, our focus will be mainly on the flow cytometric methods that we also use in the labs to be able to give you also some tips and tricks how they can be successfully applied. And as a basic outline, I will mainly focus on antigen-specific CD4 T cells in humans. Jakob will cover the antigen-specific T cells in mice, and then Tim will talk about the detection and analysis of antigen-specific B cells. So why is it now at all interesting to look at antigen-specific T cells? So while many studies looked at the bulk phenotypes and functions of these cells, it is of course important to keep in mind that the hallmark of a T and B cell is that they have antigen-specific receptors, meaning they can highly specifically recognize and react to certain antigens. But when we now look into bulk populations, those cells that react specifically to one antigen, they are typically few, but those are the cells that also integrate immune response associated changes. And then these cells, of course, also form long-term memory, meaning they remember previous immune encounters, and therefore they are readouts also for protective and pathogenic immune responses. However, these responses are also not uniform, and what is typically characterized are the frequencies, phenotype and function of these cells, clonality and affinities, and also the antigenic epitope they recognize. And with these parameters, one can then define basic mechanisms of adaptive immunity, but for example also perform patient stratification, because these parameters may differ for different patient subgroups. And finally, they can be used to develop novel diagnostics and also targeted therapies. However, the technical challenge is, of course, how can we now detect T and B cells that are specific for defined antigens? So for T cells, there are two major methods described. The first one is a direct analysis via peptide MHC multimers. And this means that you have the peptides bound to the MHC molecules, And then you need some kind of multimerization, because it has been shown that the affinity of a single peptide MHC is not strong enough to stain the T cells. However, then you can directly label your specific T cells via the TCR. However, the drawback of this method is that you need a previous knowledge of the antigenic peptides and also the MHC restrictions. And you can, of course, only analyze one or few epitope specificities. And this is difficult, especially for complex antigens. First of all, when the antigenic proteins or peptides are still unknown. And also, if you think about complex antigens that have thousands of different proteins, there are probably multiple epitopes that can elicit specific T cell reactions. And then in addition, we have the analysis of T cell reactivity. And in these assays, you typically have a mixture of different cells, and they include also antigen-presenting cells that can take up the antigen process and present it. And now when the T cells recognize these peptide MHC molecules, they become activated, and this means they start to secrete cytokines, but they also upregulate so-called activation markers. So I will come to them in a minute, and at later stages these cells also start to proliferate. And all these different parameters can actually be measured by flow cytometry. And as I said, these assays are activation dependent. The advantage is you can use all antigens, including even lysates for, let's say, complex microbial antigens, where you don't know exactly the immune-dominant proteins or peptides. And you can also, in principle, analyze all donors because the proteins you add to these samples are naturally processed by the autologous APCs in the samples. However, per definition, what you cannot detect with these assays are, for example, anergic cells or also terminal exhausted cells, because these cells do not produce cytokines anymore or do not upregulate activation markers. So this now shows an overview of different activation markers that are now widely used to detect antigen-specific CD4 T cells. And what is important to keep in mind that all these markers have also different kinetics. So for example, you have early and late markers. They have a different background level, and they are differently prone to bystander activation. So the key question is, of course, what is now the best marker for the detection of specific T cells? And also, since they have these different kinetics, what would be an optimal time point for the detection? So where activation markers were really widely used was during the COVID pandemic. And what you can see here are a few examples to show you also how heterogeneous the use of these markers is. So on the left side, it's always an unstimulated sample, and on the right side is a sample stimulated with a spike protein. However, when you look at the axis, you can see here that all kind of marker combinations are really used. And what is a problem is that specificity controls are rarely performed in these assays. So what people typically do is to take the unstimulated sample and then the stimulated sample and just put a gate around the activation marker positive cells. However, the specificity of these cells is rarely proven. And this then leads really to highly heterogeneous frequencies that have been also reported for different assay combinations. So ranging from 0.01 to up to 10% of the cells have been reported to be spike specific. And even when studies compared different marker combinations, like in this example, they in principle observed a nice correlation. However, when you look here into more detail, you can see here with the one marker combination, 10% of the cells, or up to 10%, are reactive to spike, while in the other marker combination, it's tenfold less, so only 1 to 2% are reported to be specific. So the point I want to make is that specificity controls are really needed to define the specific cells. And since we see also this huge heterogeneity, I think also standardized protocols and markers are urgently needed for antigen-specific T cell analysis. So what is the favorite marker we like to use? And of course, I'm a bit biased, but we made very good experiences with CD154 or CD40 ligand, which has been initially shown by Andreas Thiels and Mario Röderer's lab to be a highly specific marker for antigen-reactive CD4 T cells. And this high specificity also comes because there's almost no ex vivo background, and this is because upon interaction with CD40 on the APC, this marker is immediately internalized. The upregulation is also very fast, so already after 7 hours the peak time, and this means that there are probably also no major changes during the in vitro stimulation. And what is also nice, that it is co-expressed with cytokines that have the same kinetics. It is expressed on all conventional T cell subsets, but per definition, as already mentioned, we do not see it on anergic cells, probably not on terminal exhausted cells, and it is also not expressed on regulatory T cells. However, it has been shown then in 2012 by Schönbrunn and al. That the co-analysis with CD137 within this short stimulation time can be used to also detect antigen-reactive regulatory T cells, which are in this case then CD137 positive but CD154 negative. And this then shows an example how this looks like. So after 7 hours in vitro stimulation with different antigens, and you can see here there's really in this example also no background, but we see these small populations of antigen-reactive cells then coming up with the different antigens. So now I will come to another topic, and this is the detection of really rare antigen-specific T cells. Because we have seen that, for example, for virus-reactive cells, the frequencies are often rather high, but this is not the case for all antigens. And especially when we look then for autoantigens or cells in the naive compartment, the frequencies are so low that they are below the detection limit of standard flow cytometry. So to detect also such cells, we really need a high specificity of the marker and the processing of large cell numbers to be able to collect enough of these rare cells for their detailed analysis. And the solution to this is magnetic enrichment of rare cells from large input cell numbers. And in this example here from Mark Davis' lab, they made tetramer enrichment. So the cells are labeled with a peptide tetramer, and then you label your cells again with an antibody that is coupled to a magnetic particle. And what you can see here, these are the cells before the enrichment. And then you apply all your cells to a column that is in a magnetic field, so all the unbound cells will flow through. And they measured here also this flow through, so there's basically no loss of antigen-reactive cells here. However, when they elute the cells and from the column, you can see here clearly this enrichment of this large population of antigen-reactive cells. And these cells can, of course, be much better characterized than it's possible with these few cells here. So what we then basically did was to combine this activation marker detection and the magnetic enrichment in an assay that we call antigen-reactive T cell enrichment assay. And as I said, the advantage is that we can basically use any antigen we are interested in. And these can be also whole lysates, because for many of the antigens we would like to analyze, so like for example bacteria or fungi, the immune-dominant proteins or peptides are not known. And then just within this short incubation time, as I said, of 7 hours, CD4 T cells start to upregulate CD154. And this is an example how this looks then with flow cytometric counting. We get an induction of 154, but these are of course only few cells. And similar to the tetramer enrichment, when we enrich these cells really from large input cell numbers, we get of course a big cloud here of antigen-reactive cells, which then allows their detailed functional and molecular characterization. So what we also implemented in this assay is that we perform all staining steps directly on the magnetic columns. And this strongly also increases the recoveries of cells, so as you can see here, because typically with intracellular staining you have a lot of washing steps, and when the cells are on the column we avoid basically the cell loss during centrifugation times. And another advantage of this is that it also leads really to reliable or reproducible characterization of T cell subsets, because when we look here now for how many of the 154 positive cells, for example, produce interferon gamma after stimulation, you can see here, depending on the individual cell counts, we get high variability here between these replicates. However, when we perform the ARTE assay with different increasing cell numbers, you can see here that there's really a highly reproducible frequency reported. So on column staining, what does this mean? And what we typically do is to perform all staining steps directly on the column. So first of all, the surface staining, and you apply a mix of 60 microliter of surface stain to the column. It should not be less, because the columns also have a certain depth volume, so 60 microliters are important. And then we simply incubate the cells for 10 to 15 minutes in the dark, so we just put some aluminium foil on top. Afterwards, we wash the cells, and then you have to elute the cells into the tube, because the fixation is the only step that cannot be done directly on the column. Otherwise, you would fix the cells onto the column. After the fixation step, we then apply this whole fixed suspension again onto a second column. This is then washed with a permeabilization buffer. And again, the intracellular staining mix is then put directly onto the column, and cells are again incubated in the dark. And this has been described in the original paper, and also there's a step-by-step protocol published that you can find under this reference. And I think in general, this on-column staining is interesting when working with rare cell populations. So finally, I want to show you one example what we can do then with this assay. So this is an enrichment of antigen-reactive cells from really large input cell numbers, like 1 × 10⁸ PBMCs of healthy donors. And we have here one microbial antigen, MP65, but also an autoantigen and a neoantigen, so no one ever was exposed to this before. And what you can see here, when we just do original counting, there are almost no detectable cells, but following this enrichment from these large input cell numbers, we can detect cells here against all these different antigens. However, you can also see here, when we then make a phenotypic characterization, that as expected, cells reacting to the microbial antigen, they have a memory phenotype. However, for this autoantigen and also the neoantigen, we can even detect cells in the naive compartment, as well as some cells in the memory compartment. And these are obviously cross-reactive cells that have been formed to some other antigens that are able also to cross-recognize this auto and the neoantigen. So finally, I said it's important to also perform specificity controls, and I'd just like to show 2 controls that we typically also do in the lab. The first is MHC class 2 blockade, so by adding an anti-HLA-DR/DP/DQ antibody. And what you can see here, compared to the non-blocked sample, you really don't get any CD154 positive cells anymore, indicating that the induction of 154 really needs also antigen presentation. And so another control that we also often perform is a specific antigen re-stimulation. In this case, we sort out the 154 positive cells as single cells, expand these clones, and then re-challenge them either with an initial antigen or also with unrelated antigens. And as you can see here in this example, this clone highly reacts to the initial antigen. And if you do this then for the whole population, you can get an impression how many of your initially sorted cells are really antigen specific. And so with this, I'm at the end. Thank you very much, and I hand over to Jakob.
Good afternoon, everyone. My name is Jakob Zimmermann from the University of Würzburg, and following Petra's dive into human antigen-specific T cells, it is now my pleasure to cover the methods that are available for mouse antigen-specific T cells. The human immunologists among you don't leave us just yet, as you'll see that some of those methods are also applicable to a human. In this overview, you can see that the methods that Petra explained to you are applicable just as well to mouse T cells. So also here, you can re-expose T cells to their antigen in vitro and read out activation markers of proliferation. There are, however, more direct methods to identify your antigen-specific T cells, and in my part of the talk, I'm going to cover mostly peptide MHC tetramers and adaptive transfer of TCR transgenic T cells. Now let's get started with peptide MHC tetramers, as this method is available for both mouse and human T cells. The idea behind it is fairly simple. Any alpha-beta T cell receptor recognizes a peptide in the context of class 1 or class 2 MHC molecules. And the idea has been to just tag a fluorochrome on these complexes to then stain the T cell of interest. A peptide MHC to TCR interaction, however, is of very low affinity, and hence this binding is not very stable. This problem has been solved, however, many years ago by tetramerizing these peptide MHC complexes through streptavidin to now allow for the interaction of 3 to 4 peptide MHC molecules with their respective TCRs, and hence enable stable binding. Peptide MHC tetramers have been around for quite some time and been used extensively in immunology, and this table just tries to highlight the most prominently used examples, as for example the model immunology virus, lymphocytic chorion meningitis virus, or LCMV, or ovalbumin, for which both class 1 and class 2 peptides are available, and you also see that there is a fair amount of human peptide MHC tetramers available. So what are the pros and cons of using peptide MHC tetramers? On the one hand, as you'll see, in difference to the adaptive T cell transfer, with peptide MHC you're looking at endogenous polyclonal responses, which, as you'll see, is very important. It also allows for direct T cell identification without the need for restimulation, which can be really useful to assess the ex vivo phenotype of T cells, for example in the memory phase of a response. There are, however, some downsides, as for example the staining intensity can be pretty low, and thus more low avidity T cell receptors that are actually specific might potentially be missed. This problem has been addressed by using even higher order multimers, such as for example dextramers, as you can see in this figure, can really increase the staining intensity quite a lot as compared to the tetramer. Another problem has been that the detection with tetramers is quite sensitive to T cell activation, and it has been difficult to combine tetramer staining with the detection of cytokine expression by the T cells. If you are interested in that, I invite you to read this paper that has compared various methods to achieve this. So let's assume you wanted to use peptide MHC tetramers for your own research for a newly identified antigen. How would you go about that? Well, first of all, you would have to identify the peptide antigen, and there's a few known ways to do that. Classically, and especially for viruses, you can achieve this by just screening overlapping synthetic peptides. And this has been done for the P14 TCR by screening overlapping peptides in vitro assays and look for the activation of the P14 T cell receptor. While this is doable for viruses and their comparatively small proteome of just about 3,000 amino acids, this can be really challenging for more complex antigens such as bacteria, with E. coli encoding for 1.3 million amino acids. So screening synthetic peptides is really not an option. What can be done instead, however, are genetic approaches. And I'd just like to guide you through how we have done this recently when trying to identify the antigen that T cells recognize in Lactobacillus reuteri, a gut commensal. The approach has been to screen shotgun genomic libraries of our antigen of interest in vitro assays. For this, we have partially digested the Lactobacillus reuteri genome and ligated it into E. coli expression vectors. We have then transformed E. coli and induced protein expression. We have then used in vitro assays to look for the activation of Lactobacillus-specific T cell receptor expressing NFET GFP reporter cells together with bone-marrow-derived dendritic cells as antigen-presenting cells and the E. coli library expressing snippets of the Lactobacillus genome. The idea is that if the E. coli clone expresses the snippet of Lactobacillus genome that encodes for the antigen, you will see a signal. For us, this has resulted in the identification of a positive clone, and we could then complement this by synthetic peptides to really narrow down the minimal peptide that is required for antigenicity. To screen a large enough number of clones, we have done this in pools of 20 clones per well in 96 well plates and screened 8 96 well plates for a total of about 15,000 clones. A few tips here if you wanted to do this. On the one hand, the expression vector that we have used is commercially available, and there's also new frame-insensitive plasmids that give you 3 times the yield of reproductive clones that you can also use for this assay. There are, of course, other methods available, but in the interest of time, I will not go into much detail for them. There is immunopeptidomics, which means that from purified MHC molecules, peptide can be eluted and identified further through mass spectrometry. And in the last 6 to 7 years, there have been a number of approaches where antigen libraries have been combined with ways to identify positive clones in these libraries. And I invite you to have a look at these papers if you're more interested in these more advanced technologies. So now you have identified your peptide of interest. How do you get a tetramer? And it has to be noted that a really great source for tetramers is the NIH Tetramer Core Facility. It has a large selection of MHC alleles, both mouse and human, and you can get both pre-made but also custom peptide MHC tetramers. For class 2, they offer a quite wide variety of different fluorochromes, which is even greater for class 1 tetramers. Incredibly enough, this is free of charge, you just have to pay for the shipping. They do require the peptide to be shipped there if it's a custom-made tetramer. And they also require some evidence for why you think your peptide will be specific, and they also rely on feedback by customers. They also have very helpful FAQ and also have really approachable experts that can help you in designing your tetramer of interest. Also, companies offer tetramers off the shelf or custom, and an interesting feature here are UV exchangeable tetramers, where a placeholder peptide can be exchanged through UV light by your tetramer of interest. It has to be noted, however, that these are more focused on class 1 over class 2 and more on human over mouse. So let's assume your tetramers have arrived. How do you actually use them? It is advisable to use really bright fluorochromes like PE or APC, and even to use 2 fluorochromes coupled to the same tetramer to increase the specificity of your detection. As you can see in this example here from a recent manuscript where we have identified Lactobacillus reuteri specific T cells in the gut through using the same tetramer coupled to APC and PE, that gives us a really nice staining that is absent from unexposed mice. It is also advisable to optimize the staining temperature and duration for your tetramer. And the NIH Core Facility and many papers have found that this staining works mostly better at 37 over 4 degrees for approximately 30 minutes to 1 hour, but it should be optimized for every condition individually. It is also advisable to use inhibitors of T-cell receptor signaling, such as the protein kinase inhibitor dasatinib, as this avoids TCR activation-mediated internalization of T-cell receptor molecules and really improves the brightness of your tetramer staining. Ultimately, it's recommendable to precipitate aggregates by a quick spin at full speed for 1 minute. As you can see from this paper, that really gives you a much cleaner staining. And as always, and especially for tetramers, it's absolutely paramount to include viability dyes and the dump channel to really have your staining as specific as it could be. So to give you an idea of a typical staining protocol for us, we isolate T cells from the mouse gut, for example, and we stain them with 1AB tetramers in PE and APC, diluted 1:400 in IMDM with dasatinib. We take a quick spin and stain them with the supernatant of the tetramer mix. We incubate cells for 1 hour at 37 degrees in a cell culture incubator and use 96 well plates for that. Following the tetramer staining, we do successive stainings for viability, surface antibodies, we fix the cells and also stain for transcription factors. And just to give you a vignette of how well the combination of tetramer with transcription factors work, you can see here the co-expression of tetramer or the co-detection of tetramer together with transcription factors ROGMA-T on the left, BCL6 in the middle, and KR67 on the right. Now, of course, it is also critical to really determine that your tetramer staining is specific, and there are several ways to do that. On the one hand, the NIH Core Facility always adds to your card automatically the human CLIP negative control tetramer. CLIP is the class 2 associated invariant chain peptide that binds really well but should not reveal any staining. It's also advisable to have biological control, such as, for example, unexposed mice, and you can use cell lines that express your TCR of interest or an irrelevant TCR to make sure that your tetramer staining is nice and specific. Second part of my talk, I would like to focus on the adaptive transfer of T cells as a method to detect mouse antigen-specific T cells. The principle is fairly straightforward. One transfers a monoclonal population of T cells that are all specific for your antigen of interest. These can in the host be discriminated by a congenic marker, such as, for example, CD45.1 or 0.2 or CD90.1 or 0.2. Then one does the immunization or colonization or whatever and can then interrogate the specific phenotype of the antigen-specific T cell, such as, for example, here, transcription factor staining. There are several TCR transgenic mouse lines available, and they have been used for many decades in immunology. And again, I would just like to highlight very prominently used ones, such as, for example, the P14 or SMARTA TCR transgenic lines specific for LCMV, CD8 and CD4, or OT1 and OT2 mice specific for ovalbumin. Now for TCR transgenic transfer, what are the pros and cons? On the one hand, one has to say that the detection is really robust, as CD45.1 and 2 are really brightly expressed surface proteins that also makes this assay compatible with reactivation and cytokine staining. It also enables you to actually use genetically modified mice as donors, as for example in this paper from David Massapus' lab, where people have compared TCR alpha flux and wild-type T cells after adoptive transfer and could then really interrogate the function of a gene only in the T cells. There are some downsides, however, and these two papers from 2007 have found that the adoptive transfer of high numbers of monoclonal T cells really impacts the phenotype of the T cell population when compared to lower numbers or endogenous responses. For example, for CD8 cells, it was shown that they then differentiate more towards memory and less toward effector cells, as shown by increased expression of CD127 and reduced expression of granzyme B. And for class 2 and CD4 T cells, it was shown that the interaction of a specific TCR with a peptide led to differences in the differentiation of Th1 versus TFH cells, even if all these peptides were expressed by the same pathogen, that is Listeria monocytogenes. So one just has to be aware of these limitations. Now let's assume you wanted to do adaptive T-cell transfers in your lab. How would you go about this? First of all, you would have to identify a T-cell receptor. This has classically been done by taking infected mice, as for example with LCMV, taking the T cells from these mice and propagating them in vitro with infected macrophages. Then you could do limiting dilution and identify really individual T cell clones, test their reactivity, sequence their TCR, and this is how, for example, the P14 T cell receptor has been identified. This has also been recapitulated more recently with T cells specific for the gut commensal Akkermansia muciniphila, with the addition that CD4 T cells were fused with lymphoma cells to generate immortalized hybridomas that could then be sequenced and tested for their reactivity. Now we have done it in a slightly different way that circumvents the need for generating T cell lines and clones, and that is single cell RNA sequencing. The advantage is that you combine information of clonotype and phenotype. The clonotypic information will tell you whether your T cell receptor candidate is expanded in exposed mice, whether, for example, it's even public, so present in more than one exposed mouse, and it's unique, so present in exposed but absent from unexposed mice. This can then also be combined with phenotypic information such as Th17 Treg and so on. In the case of our recent manuscript, we have also combined this with the antigen reactivation, so we sorted for T cells and performed single-cell RNA sequencing after in vitro re-exposure to the antigen. Now that you have identified a T cell receptor candidate, you would want to make sure that it's actually specific before going for in vivo experiments. And the way this is done is that you express your T cell receptor in a reporter cell line, you co-culture it with antigen-presenting cells and antigen, and you read out T cell receptor activation. This is most commonly done in reporter cell lines that use some way of NFET-driven reporter gene expression, such as GFP, as in our example. This is what this has looked like for us. So here we have looked at NFET GFP expression after co-culture with only BMDCs, where you see for these 3 T cell receptor candidates, there is very little expression. We have a positive control, whereupon we see lots of NFET GFP in the middle row, and in the lowest row you can see that when we use heat-killed lactobacillus particles as antigen, we can nicely validate the specificity of 2 of thus tested T cell receptors. A few tips here. Many of those lymphoma cell lines lack these 3 CETA subunit, but you can reconstitute this with a plasmid available at AdGene. In terms of antigen-presenting cells, if you have complex antigens such as bacteria, lysates, it's advisable to use bone marrow-derived dendritic cells that can be derived from mouse bone marrow following a week's culture with GM-CSF. If you have more simple antigen, such as protein or peptide, you can use immortalized B-cell lines, as in the reference I'm showing here, that are really capable for presenting such more simple antigens, but are poor at presenting bacterial particles, for example. And another tip is that the NFET luciferase cell line is actually available commercially. So you have identified and validated your T cell receptor of interest. How do you get to the final stage of expressing it in primary mouse T cells? And in my last slide, I'll guide you through the 3 major steps to do that. Classical TCR transgenic mouse line, such as P14, for example, this has been achieved by injecting the pronuclei of zygotes with expression cassettes for TCR alpha and TCR beta. These then integrate into the genome randomly and often at multiple copies. While these carry their own promoter regions, the expression will still depend where in the genome these integrate. This does achieve germline transmission, and it's fairly fast and of intermediate cost. A new, more physiological method has been introduced by Kilian Schober and Dirk Busch a few years ago, and that is called orthotopic TCR replacement. What this method achieves is that it integrates the transgenic TCR into the endogenous T cell receptor locus, thereby ensuring physiological regulation. I'll spare the details of how this works and I'll invite you to read their paper, but it's important to notice that this works both for human and mouse primary T cells, but also stable TCR transgenic mouse lines can be generated with this. This is a targeted mutation. Generating mice like this will involve some more time and cost as compared to a classical transgene. Now, the last and most doable approach are TCR retrogenic mice. As the name suggests, this involves retroviral transduction with the TCR. Classically, one isolates hematopoietic stem and progenitor cells identified by SCA1 and spin infects them with the TCR expression construct. One can then irradiate recipient mice and reconstitute their bone marrow with the transduced hematopoietic stem cells. This can be done really quickly, so in a matter of 5 or 6 weeks, these hematopoietic stem cells then give rise to mature naive T cells that can be isolated for adaptive transfer. It's also fairly achievable in terms of finance. However, of course, it also has random integration of the retrovirus into the genome, and the expression will be somewhat locus dependent, and there's no germline transmission, so this has to be done over and over. But I'd like to stress that this is really a method that can be achieved even by non-experts. A few tips here: the PMP171 retroviral plasmid is available at Adgene, and also platinum E cells to package your retroviral particles are commercially available. If cloning is an issue, it can be outsourced to companies too. If one wants to avoid endogenous recombination and potential dual TCR expression, then RAG deficient mice could be used as donors. Before closing, I would like to shout out to a new technology, and that is the generation of multiplex TCR retrogenic mice enabled by fluorescent barcodes called flow codes. If you're interested in this, I invite you to look at the webinar given by Adrian Liston that has recently been uploaded on the ISAC YouTube channel and is now freely available. In this, individual T cell receptor expressing populations can be identified by a fluorescent barcode that can be then read out in flow cytometry. With this, I'd like to close my part of this talk, and I hope I could convince you that detection of mouse antigen-specific T cells is A, worth it as it really zooms in on the relevant cells, B, really doable even for non-expert labs, enabled by available resources, published protocols, and approachable experts, and C, achievable through several different methods that all have their pros and cons that one should be aware of.
Hello everyone, my name is Tim Rollenske, and for this third part of the webinar we're going to switch from T cells to B cells, but the goal of the webinar stays exactly the same. So we want to enable PhDs and postdocs, in this case now working on B cell immunity, to go beyond their bulk analysis towards analyzing their questions at single B cell level and also the monoclonal antibody level. And I will present mostly methods that we are using every day in my own lab, but I will also mention some alternatives on the way, so that you will be enabled basically to find the best solution to your specific question and your specific project. Okay. For the beginning, I wanted to show you our strategy to go beyond bulk immunity. And therefore, the first step is always to identify antigen-specific B cells. And we work on the mucosal antibody response, so we work with humans and with mice. And there we work with intestinal tissues most of the time. And we work with germ-free animals that we then can colonize at different time points with different bacterial consortia. So after you identified your antigen-specific B cells, their location, their phenotype, and their frequency, what we do is we isolate those cells and then perform FACS sorting. Then we perform single-cell IG gene sequencing to receive the B cell receptor genetic information or the B cell repertoire. And then we use custom code or published code to analyze those repertoires. We look at their mutation status, public clones, etc., etc. And then if we are interested in a specific population of clones, then we have developed methods to produce monoclonal antibodies in a high throughput fashion. So now we are at around 3 weeks for 100 antibodies. And those ones then you can test for their reactivity to whatever antigen, bacteria, or self-antigens. And if you then find interesting clones, you can test them in vitro and in vivo functional assay. And I will just give you one example where we actually went beyond bulk immunity. So we took germ-free antibody-deficient mice and we colonized them with a single E. coli strain for 3 days. And then after the 3 days, we injected 400 microgram monoclonal dimeric mouse IgA. And what you can see at the bottom is that this antibody is quickly cleared from the serum and then appears after 6 to 12 hours. You see a peak in the feces. So this allows us now to study the functional effect of this one clone in the mouse intestine. Okay, but maybe you are already only interested in the step 1, so in the location, the phenotype, and the frequency of your cells, and then you don't need to go down that road with all these different methods. So you can only do, for example, an ELISpot. That might be sufficient. Or in your project, you basically just require a monoclonal antibody, and in this case, generating hybridomas or using a nanobody or even an AI binder might be already sufficient to answer that question. Okay, but the first step that you need is basically to find a good FACS sorting strategy if you want to identify those antigen-specific B cells. And therefore, I thought for mice and for human, I provide you one. And this one comes from this nice paper, the reference at the bottom. And it's a very standard flow gating strategy where you can gate on germinal center B cells and also non-germinal center B cells at the end. And they have identified the most of the antigen-specific B cells that are actually sitting in this class-switch non-germinal center B cell population. And one important aspect I would like to mention also on here is that actually, if you are interested in plasma cells, then you run into a problem with the antigen-specific staining, because fully differentiated plasma cells, they do not express surface BCR except IgA-positive plasma cells, maybe the reason we are working with those. And then you need intracellular staining. And there are protocols where you can perform intracellular staining and still perform RNA sequencing at single-cell level. But I haven't found any information that it can be linked with antigen-specific staining, and then you retrieve the BCR information. So to my knowledge, these experiments are very often fishing experiments where you basically generate 100 monoclonal antibodies and you basically need to test all of them to find certain specificities in there. So for humans, this looks similar, so very standard gating strategy. And then if you're interested in, this is peripheral blood now, so mostly you would be interested in plasma blast, so CD27, CD38 high, and then, or in memory B cells or atypical memory B cells, so CD27 positive, CD21 positive for classical, and then CD27 negative, CD21 negative, and then mostly class switched for the atypical memory. And you can see from there, in this example, they gated on CSP, which is an antigen on the malaria parasite. So if you have then your gating strategy, your next step would be to gate on your antigen-specific cells, and there you can use different staining agents. And the most used and classical one is you take your protein of interest and you just couple it to any fluorophore. Here I show you two examples, again the CSP and then one example of HIV GP140. And in our lab, we mostly use Alexa 647 or biotin. Sometimes we also use FITSI. So those are the ones that work best in our hands. And of course, there are also other possibilities to stain antigen-specific B cells. So you can directly use fluorescent proteins like PE or APC. This is a very elegant method. We have used bacterial glycans that were biotinylated to identify human memory B cells. And also people have used whole bacteria to isolate antigen-specific B cells. So this is dependent on your project. Here I wanted to give you also some pro tip. So of course you need some dye only or unbiotinylated antigen control. What is very elegant is always a double dye staining. This increases the specificity a lot. For this whole bacteria, blocking may be required. And also we try to use the lowest volume of staining mix possible, because sometimes there are some aggregates in this mix. So the more mix you use, the higher your background actually gets. So positive versus negative control, you should definitely use the same amount of staining mix there. Of course, people trying to go one step beyond. So there you can stain actually multiple antigens at the same time. So here's one example where they used tetramers to attach a protein to it, and then also the tetramers have a barcode. And then they stain memory B cells with it, and you can see that you find some that are binding just to the one protein, whereas others exclusively bind to the second one, and some are actually cross-reactive. And this you can sort, and then you can perform CYTO and then later bioinformatically sort which cell were actually binding to which antigen. And we are very interested in this. So I want to start a challenge. So how many antigens you can actually stain in your project, and if you go beyond 3, so then I'm very happy if you send me an email. Yeah, because we are very interested in this kind of method. Okay, after the first step, the next one, so you sort your cells, and then the next one would be obtain the BCR information, or to do single B cell sequencing, and to link basically the BCR reactivity to the IgG genetic information. You can do this with bulk, but if you want to generate monoclonals, I highly recommend to do a single cell technique here. Because it's basically impossible to generate monoclonal antibodies from this bulk sequencing data. And to do so, to get the BCR information, there are two main methods. So the first one is that you basically sort a single cell in each well, then you perform reverse transcription and then IgG PCR, and this one you can sequence by Sanger. And this method has been used for over 20 years now. It's relatively cheap and is a robust method, but it of course has low throughput. What we are using now most is actually that we sort all the cells into one tube, and then we perform a drop sack, and we include a cell hashing process, so we can bioinformatically sort which cell belong to which donor. And this has, of course, a high throughput. And con would be that the cell hashing process and also this FACS requires more prior testing. Also here I wanted to give you some pro tips. So you should buy the cell hashing antibody clone, so the same clone in your favorite color, and titrate it. We actually add the hashtags before the sorting, so this works very nice. and you should test the FACS conditions and test this beforehand, that the cells stay intact with your sorting strategy. And of course, there are alternatives to DropSec. So we use 10x genomics here and Cell Ranger to retrieve the BCR information, but there are, of course, other platforms. And with this full-length VDJ sequences that you get from both methods, we are feeding this into the incantation suite. And there are many webinars about this. You can find this at incantation.org. There you find all the information, how to install this, which packages can be used, and I will not go into further detail into this analysis. However, I want to give you a small pro tip, which antibodies, so how could you select actually some of the antibodies of interest. So, of course, you should look out for hallmarks of positive selection. So, if your antigen-specific B cells have an Ig gene bias, if they have shared somatic hypermutation, or they show signs of antibody affinity maturation, like a high somatic hypermutation count in their V genes, or if they are massively clonally expanded. This can all be hints towards antigen reactivity. And when you want to express a monoclonal antibody, most of the antibodies and most of the groups are expressing antibodies as human IgG1, but of course also mouse IgG and different human IgG subtypes are possible. And this is all summarized in the reference 1. So there, if you're interested in this, just go there, and we basically provide the newest method how to clone these monoclonal antibodies and how to express them. And in reference 2, you find then the information how to produce mouse dimeric IgA, which is the isotype that we are mostly using in the lab. But the principle between these two methods is basically the same. So because you start with the PCR or with the DNA synthesis of the heavy and the light chain insert, so full-length VDJ, this you clone into, in our case, IgA, Ig kappa, Ig lambda expression vectors, and then together with an IgJ vector, because we want to have dimeric IgA, you transfect HEC293F cells. And from there, you can perform affinity chromatography if required, and also you can perform size exclusion chromatography, which is sometimes required if you work with dimeric IgA. Okay, and these, of course, then you can test for antigen reactivity and specificity. There are, of course, some alternatives. This is here in reference 3 and 4. These are methods where you force B cells ex vivo to secrete antibody, and this you can achieve by single B cell culturing and also by B cell immortalization. And if you're interested in this, you should check out these two references. So at the end, I wanted to talk about the most important question. So how do you actually select high affinity antibodies? And here you see a correlation of the MFI value that you have in flow cytometry versus the actually affinity of an antibody measured in this case by surface plasmon resonance. So here these antibodies were sorted against the circumsporozoid protein, which is an antigen on the malaria parasite, and then the affinity to an epitope, a dominant B cell epitope of this proteins was tested. And you can see that normally if you have a high affinity, you also have a high MFI in the flow staining. However, a high MFI in the flow staining does not guarantee that this antibody derived from this cell actually is of high affinity. So, meaning this is another example why it's really important to go beyond bulk immunity, to really go to the monoclonal antibody level and to show that these monoclonals are actually of high affinity. And with this, I would like to close. And we thank a lot of people. Here you also can find our contact information if you have questions. you're always welcome to contact us, and we are happy to answer now your questions.
Thank you very much for the wonderful talks. Great series of talks, and honestly, the multimers and all have always been something that was near and dear to my heart, specifically the CMV. I wanted to really get a handle on the antigens. When I think of really more of the CD8 responses, but really CD4, CD8, these were always peptides that we were working with. But it sounds like a lot of you were working with whole proteins or had different approaches to looking at that. What are the actual antigens that you typically use? And I guess Petra, do you want to go first or?
Yeah, thanks. I can go first. So we are also mainly interested in microreactive T cells, and there we have exactly the problem that the proteins and peptides are not known. And when you look, especially then also for something like a fungus, they have 6,000 different proteins, and then you can calculate how many possible epitopes that would be. So that's the reason why we start with a lysate, and this is also the reason why we mainly use this activation-dependent methods, because in this way you basically get the whole response, and then you can start to dissect it, right? So if we sort out then the activation marker positive cells and re-challenge them with individual proteins, we try to figure out what they recognize.
Anybody else? Jakob?
Yeah, so we've looked at the T cell response to gut commensal, also complex bacterium, Lactobacillus reuteri. And funnily enough, we've used two different methods to identify specific T cell receptors. And two of thus identified T cell receptors recognize almost an identical epitope from the same protein, and that's a surface kind of attachment protein. So it seems that even if there's this large diversity of proteins, some of them are really more immunogenic than others, it seems. And for us, that was really helpful because we could then devise those TCR transgenic adaptive transfer methods, and those really also get activated strongly when we colonize mice with this bug.
And Tim, how about with B cells? Anything? Is it?
Luckily for B cells, we don't have this problem because we can use glycans or proteins as we like and peptides, and we can stain all of these actually.
Yeah. Definitely hard on the T cell. You were mentioning adaptive transfer, and I was just curious. It's sort of a side question, but when you were showing giving different doses of adaptively transferred T cells, that you swayed the response from more of an effector response, I think, more to a memory response. And just from the immunological perspective, I wondered why that happens. What is that? What's going on immunologically in terms of that?
Yeah, I think that's not entirely clear. I think it's a bit, since these are all monoclonal, they might be competing for the same kind of self-peptide MHC synthesis for their homeostatic proliferation kind of signals. And in the papers I was showing for CD4, I think the reason is really the affinity of peptide MC to the TCR and mostly the K-off rate that determined whether these would become Th1 versus TFH cells. So really there is a bias from the way the TCR recognizes peptide MHC to how those T cells differentiate. So I mean, one just has to be aware of that kind of.
It's going to happen, yeah. But I mean, is it just that you think you're running out of antigen, and so they're moving more towards a memory response, or?
I'm not so familiar with those CD8 papers, to be honest. I think the practical takeaway is just to really not exaggerate with the amount of antigen-specific T cells, so maybe not go for 500,000, but limited to something like 10K. I think it's been shown that the seeding rate is like 5%. So maybe if one transfers 10K, then you have something like 500 cells ending up, which is not too far off of the physiological frequencies of about, I think, 100 cells with a given peptide specificity for a given epitope.
I've got other questions, but I wondered if the three of you had any questions for each other or had anything that you wanted to ask about each other's presentation.
I have questions. No, especially Petra, for your human analyses. I'm always wondering about the role of the antigen presenting cells. And of course, when you work with blood, I guess that's pretty standardized. Like you have your DCs, macrophages, B cells. But what happens, because you also did tissues, right? Like biopsies from patients. Do you consider which kind of APCs you have in there, or do you standardize that by using the blood from the same patient, or how do you deal with this?
Yeah, it's a great question. It's often difficult to do, right? Because you get limited amount if you get a small biopsy, and then you're happy with what you have inside. But it's a great question because also different APCs might have different processing capacities, might cut different epitopes. We have not gone deep into that. We are still scratching at the surface, and that's what I said. I mean, we are looking for lysate-specific responses. And there's still much to discover, but as soon as we come closer to individual antigens and epitopes that are recognized, I think this would be something to revisit. Also, if the type of the APC has an impact on how these epitopes are generated.
And I have a
Which,
question.
go
I
ahead,
would
Tim.
also have a question to Jakob.
Okay.
Jakob, in your opinion, so how many CD4s do you actually need to transfer in terms of to get the seeding to the gut? So what is the best number to
We
transfer?
haven't done this kind of experiment. I mean, people have transferred individual T cells, like single ones. There's this work from Bush's lab where they've generated a congenic matrix, like using all possible combinations of 45.1.2 and 90.1.2. And they have a pretty good recovery of just single T cells. And then they made this connection between how they then differentiate towards memory and effector. So this is possible. But on a practical level, maybe that's not what every lab can do on a daily basis. So what we typically do is like something between 10 and 30K. And we then end up after this colonization with like 1 to 5% of CD4s in the kind of peak response. So that definitely gives us good enough numbers to look at their phenotype to isolate them for RNA sequencing and things like that. Tim,
Okay. Well,
like when you were saying PE and APC, does that mean you use these as antigens, like you immunize a mouse with PE, and so it then directly stains with PE as well, the B cells?
Exactly. Yeah, this is what they did in this paper. Yeah, so they add the PE CFA, and then you can track PE reactive B cells, and this quite practical.
That's correct. Yeah. And
that's
another
cool.
question for you, Tim, what kind of specificity controls do you usually do? Because proteins, I mean, they're sticky, they could just unspecifically bind, right? What do you think is important to make sure your fluorescently labeled protein really targets only the specific BCR and not something else?
Yeah, we mostly do dye control or unbiotin-related protein controls. And the best one is actually if you do double dye, So if you have FITC and Alexa 647 staining, and then you see a little bit background basically for the single reactivity, and then if you sort those for double reactivity, I have seen a high frequency of antigen reactive monoclonal antibodies that were received from this population of B cells. So this double dye is kind of the best thing that you can do. Yeah.
And one last
Yeah,
question
that's interesting.
For
I thought that was an interesting approach. I agree. Yeah, with the dual fluorochromes. Go ahead.
There is because for cytokines, there has been the same problem, right? That you cannot really stain T cell cytokines and then do single cell or whatever because you have to fix. And the solution has been the cytokine secretion assay, where you have this matrix where you label T cells with an antibody that covers like CD45 and the other arm binds your cytokine, and then you let them secrete in vitro. Can you do that with plasma cells as well, like generate a matrix and then have them secrete the antibodies to capture those that then specifically secrete the specific antibody to then sort and do sequencing or whatever?
Yeah, they're basically methods that circumvent this process that you first need to get all the cells as a repertoire, and then you clone specific antibodies. So these methods have other pitfalls, but there are methods that you sort B cells and then you generate them to, you differentiate them to plasma cells in vitro, either by sorting them and then by culturing or by immortalization with EPV. And then you can directly harvest from there, do an ELISA, and then you can only look at those wells where your specific antibody is. But of course, this is also not unbiased, no? So you're not looking at all the cells in your response. I've seen that there's actually a question in the
Yeah. So Tim, yeah, I thought that was really interesting at the very end where you presented the data where the MFI didn't correlate really with the affinity, and I wondered if you could really speculate on what that was, and then to the specific question, if there are any other markers on B cells that would maybe a better way of looking at affinity, but that we could use by flow.
So I was so I think the markers, so that the with high MFI you get around 50% are high affinity, at least in this data. I found this already quite good, I have to say. So I'm not aware of any better marker. So I would speculate that the amount of BCR is different on the surface, and then it depends if I have multivalent antigen or not. So we tried this also with glycans that are repetitive. So they have repetitive epitopes, and they are also different in size. And there we see sometimes that the MFI then stops correlating with the affinity of the monoclonal antibody, and that it rather works for single epitope proteins, this correlation. Yeah. So I would speculate most probably is the amount of BCR that I have on the surface.
The B cell receptors there. Yeah, interesting. All right. Well, we have gone over, and Anna's going to be mad at me for going long, but I really do want to thank you all very much. So on behalf of the ISAC community, we'd really like to extend a sincere thank you to everyone who joined our presentation today. It's been a pleasure connecting with all of you, as well as with our speakers. If you're interested in exploring more of the learning opportunities offered by ISAC, we encourage you to check out our archived CYTO U recordings and on-demand webinars. These resources are available to you anytime. At the end of today's, you will receive a short survey. Please take a few minutes to fill it out. Additionally, today's webinar has been recorded and will be accessible on CYTO U very soon, so feel free to revisit any information at your convenience. Be on the lookout for announcements on our next webinar on April 14th, featuring past recipients of our Exceptional Student and President's Award for Excellence. Thanks again to everybody for joining us. Enjoyed it very much. Bye now.